So we're going to move on to the panel. This is addressing global health diagnostics and the limited diagnostic capacity in low resource settings, which undermines the effectiveness of global health programming and can drive the emergence of drug-resistant infections. And so this panel is going to focus on new frontiers in low-cost point-of-care diagnostics in the area of global health. And we'll also discuss innovative university and industry advances as well as critical gaps in the field. And what we're going to do is have the moderator, who is going to be Rupa Ramamurthy from the BBGH. And do you want to come up? And then who will be introducing and moderating the panel with Daniel Fletcher, from professor in bioengineering, and Professor Luke Lee, also in bioengineering.
Hello, can you all hear me? Yes. Okay, great. Diagnostics is only accounts for about 4% of the total investment in health. But however, about 60 to 70% of all decisions are affected, healthcare decisions are affected by diagnostics. And this 4% figure in spending is also true in global health. Now, there are a number of challenges for diagnostics for neglected tropical diseases, TB, malaria, and HIV. One is the regulatory challenge. There is not enough of quality assurance. You have a number of diagnostics, and not all of them are of equal quality or reliability. WHO has a pre-qualification system, but the pre-qualification system is much behind that for drugs and vaccines. And going through FDA approval is expensive, and sometimes companies go through the EU CE mark. Then the target product profile for these diagnostics. There is often no power or very intermittent power, lack of clean water, high temperature, humidity, and also someone who is not very skilled, so that the diagnostic is easy to use. The other concern is, where do you want the diagnostic? Do you want it in a central laboratory, peripheral, or point of care? And also, sometimes the diagnostics have to be country specific. For example, if you have a fever panel in a country like India, you may want to include dengue on the panel and may not be in some other country. Then the market issue. Who pays for the diagnostics? What is the price point, and how is it going to be delivered to those who need it? For example, in vaccines, you have Gavi that gives advanced market commitments for vaccines, and as yet, there's nothing similar in diagnostics. And then some of the big diagnostic challenges. One is a fever panel for children under 5 to distinguish between, say, pneumonia, malaria, dengue, because otherwise children are just given anti-malarials, and that's causing drug resistance. Then in case of Chagas, how do you find cure for chronic Chagas that's affected the heart muscles? And you need a diagnostic to say that the cure is working for chronic Chagas. And the other thing is for TB, using samples other than sputum, because for young children it's very difficult to extract sputum. And also, when there is HIV and TB, sometimes the tests are not sensitive enough. I'm from BioVentures for Global Health, and what BioVentures has done in the diagnostic space is Priya Mehta put together the diagnostic roadmap in May 2010 for neglected tropical diseases, HIV, malaria, and TB. There is the Global Health Primer. There is the paper copy, but it's also online. And among other things, it has diagnostic pipelines for all the diseases, along with the stage of the diagnostic, which is updated every 3 months. And then the new program, which I'm mainly involved in, is WIPO Research, where you have a number of the big pharmaceutical companies have joined in with along with the World Intellectual Property Organization, and BioVentures for Global Health is the partnership hub to bring together the various partners to accelerate the development of drugs, vaccines, diagnostics for neglected tropical diseases, TB, and malaria. And as of now, the database is more focused on drugs and vaccines, but we want to emphasize diagnostics also. And there is a great importance in frugal engineering, both in the developed and developing world. And Professor Luke Lee would be talking about universal sample preparations. And then another interesting thing is that, for example, in India, more people have access to a cell phone than a clean toilet. And Professor Daniel Fletcher will be talking about cell phones and microscopes for diagnostics. So I'd like to invite Professor Luke Lee and Professor Daniel Fletcher to...
All right, thanks very much for the introduction, and it's a pleasure to be here today. I want to tell you a little bit about a project in my lab that's focused on trying to do microscopy in the field using simple and low-cost microscopes. Now, the research into global health problems and into solutions for global health problems have been greatly aided by modern microscopes. So this is an example of a very fancy and expensive microscope that does a wide range of different things. This is the kind of microscope that we like to have in lab to allow us to do imaging of a multitude of different properties. But in many settings, in order to take an image that's useful, you don't need these sorts of capabilities. And so we've been working on a technology that allows you to do imaging in remote locations using cell phones. And the basic idea is to build something smaller, more compact, that's almost actual size, and allow you to interface with the image and with the sample and sending the image using what has been already advanced a great deal by handset manufacturers. So we're trying to take what's been done and apply it to imaging capabilities. Now, the reason to think about imaging still is that being able to see the cause of disease is still one of the most direct ways to identify and to track the development of that disease. So here are just a few examples of where microscopy is used for either screening or diagnosis for infectious agents. And while the grand goal, I think in many global health diagnostics, is to have something like a rapid diagnostic strip that you can apply a sample to and know immediately what it is that you have or what subspecies you might have. Right now, microscopy, being able to visualize those disease agents is critical. And so we want to try and move that technology into the field as development of other diagnostic technologies continues. It's worth mentioning that microscopes weren't always enormous, and they weren't always very expensive. They started out very small, very compact, and very portable. This is an example of one of the earliest microscopes by a gentleman named Antony van Leeuwenhoek. He produced microscopes that consisted of a single lens that he polished himself and mounted on a metal plate and peered through. And it was using a device like this that he discovered the first microorganism. Bacteria, giardia, things that were moving in samples that he found around him. Now, microscopes have expanded in size and capability due to a wide range of different contrast mechanisms that are now possible. And many of those have allowed for identification of specific disease agents. Several years ago, in an undergraduate class that I teach on optics and microscopy, one of the projects for the semester was being able to use the cameras that were just emerging on cell phones. I don't know if you can remember that far back when cameras were not on cell phones, but there was a time when a cell phone was just for talking. When they emerged with cameras, these cameras were very low quality. They couldn't do much, but the possibility of being able to use that imaging sensor to take pictures of small things, not just big things, was the subject of that project. And what we identified in the course of doing this work was that, yes, cell phone cameras are good enough to take images, magnified images. And what got us excited about the project even more was the capability of even sensors from that day and age to be able to take fluorescence images. And fluorescence microscopy has become a workhorse for a range of different molecular diagnostics as well as disease agent diagnostics. And so building on that basic and simple concept, we've built a first device and have been trying to expand the range of capabilities, all piggybacking off of the capability of the handset itself. And so the basic concept is this. We have the health worker or an individual who's got the ability to capture images at the site with the patient. You have the ability to do some amount of image processing and analysis on the phone itself based on the computational power of that phone. But you also have the ability to transmit. And you can transmit both for record-keeping purposes, for second opinions, or for the ability to have additional diagnostic information derived from those images. So what we've done since our initial device is expand the range of types of samples that we can work with. This is an example of a more recent iPhone-based device, which consists of a set of lenses, an LED illumination, and can do bright field imaging of stained blood or unstained blood, and a range of other samples. This system can also be made into a fluorescent system. But there are other things other than single cells that are worth imaging. If any of you have had ear infections, being able to visualize that your eardrum is inflamed is something that is a very common diagnostics in a pediatrician's office or in a general practitioner's office. Being able to do that on a cell phone is something that we found is equally simple, simply by adding a lens onto the imaging system. And more recently, the ability to image into the eye and look at the retina has become possible with, again, the same exact phone. So we've expanded the ability to do imaging for different disease types, but it's all based on, right now, an iPhone, but the concept is very general. Other phones can be used as well. So we're very interested in taking advantage of the increasing number of pixels on cameras, the increasing sensitivity of those, the ease of use of the touch interface to be able to do more and more imaging that's relevant to global health. So here are a few of the application areas that we've explored. Blood smears, malaria, tuberculosis. We've done some work in education and imaging to figure out what those white powders might be, as well as agriculture and trying to do basic science where field work is often prevents you from taking your large microscope with you. So I'll give you one example of where we're trying to move beyond simply device development and actually test whether we can put devices into settings where they're actually useful. And that is an example in Vietnam. So together with collaborators at UCSF, we're participating in a project supported by TB Reach that is trying to improve the ability to diagnose tuberculosis. And the method for doing that is twofold. One is using a very modern technology, the Gene Expert, that can detect drug-resistant tuberculosis. And the second aspect of this project is to try and push the ability to do the screening down to lower-level health clinics. So right now, the lower-level health clinics, the commune health posts, don't do diagnostics. But if they had a microscope imaging system that was easy to use and that had the ability to identify or help to identify what disease agents are there, then those clinics can participate in the initial round of screening and then push only those patients which have been found positive to the larger clinics for initiation of therapy. And so that work is ongoing, and several students who are participating in that project are here today. So I'll end with this again. The goal is not to replace these microscopes or put these large microscopes into the field, but rather to take advantage of imaging, which is becoming ubiquitous, and see if we can apply that to important problems in global health. And there's some of the people who've done a great deal of work to advance this project on multiple fronts. And it's been a wonderful collaboration with folks at UCSF to identify where can we take advantage of a device once we've built it. I think I'll stop there.